name is Oliver Fien. I'm director of the West Coast Metabolomics Center. And so we have a monthly seminar series, and this seminar series is now focusing on data science. So we are supported by the Genome Center, specifically the focus group, Data Science and Metabolism. So that's where you are, sitting right now. We will have more and more focus on data science because, well, we acquire more and more data. And as we acquire more and more data, well, we have to figure out what to do with the data, don't we? Right? And for that, we typically used our intelligence, but it turns out that our intelligence is not intelligent enough. So we now rely on more intelligence from outside sources. And that's also what our honored speaker today will talk about. So Professor Olga Vitek is the Raymond Bradsteed Professor in the Curie College of Computer Science at Northeastern University based in Boston. She's also director of the Barnett Institute for Chemical and Biological Analyses and former Cy and Laurie Sternberg Interdisciplinary Associate Professor. So her education, she did a PhD in statistics at Purdue University, a master in mathematical statistics at Purdue University, and a bachelor at University of Geneva in Switzerland. So she became professor at Northeastern University in 2016, was faculty at Purdue from 2006 to 2016 in the Department of Statistics and Computer Science, was before a postdoctoral fellow at University of Washington, Department of Genome Sciences. Jeez. And her research focus is on statistical methods and computational tools for mass spec-based proteomics and metabolomics, so that's new. Quantitative proteomics, label-free, isobaric labeling, targeted MS, single-cell proteomics and metabolomics data analyses, machine learning for biological data integration, open source software, we like open source software, including Cardinal. I hope you will talk about that. She had multiple honors and recognitions, including the Chan Zuckerberg Essential Open Source Software for Science Award, the Gilbert S. Omen Computational Proteomics Award from the US HUPO organization, the Excellence in Research and Creativity Award from Northeastern University, and the National Science Foundation Career Award. Professor Vitek is a fellow of the American Statistical Association, senior member of the International Society for Computational Biology, and an elected member of the Council, HUPO Council, and its US Board of Directors. She is editor of multiple journals and advisory boards, and president of the Boston chapter of the American Statistical Association. All right, without further ado, I welcome Professor Olga Vitek. Floor is yours.
Thank you so much. Thank you for the kind invitation and for the introduction. So indeed, I am trained as a statistician, and this type of interdisciplinary career path, I guess, is increasingly common now for anybody interested in data science. I was trained as a statistician. I did my postdoc in field related to mass spectrometry, and now I'm a professor of computer science. So it's really kind of moving from one field to the next. But what remains unchanged throughout that is my interest in bringing statistical rigor and analytical rigor to the interpretation of mass spectrometry-based experiments, especially when we talk about quantitative aspects, maybe more so than just identification. And so that's what I would like to talk about. So my lab in general has three directions. One is we do a lot of work in proteomics. The other would do a lot of work in mass spectrometry imaging, and that's kind of the topic for the discussion today. And then the third would try to go beyond just interpreting the raw data, but actually trying to make sense of the underlying biology as well. So we have three parts, and I will really talk today about mass spectrometry imaging and Cardinal. And so what I would like to do is to start just with the overview and then dive deeper in a couple of topics. And the first is Cardinal. So it is a flagship software tool from my lab, which was started, I would say, 15 years ago or so, more or less, by Kylie Bemis. Kylie used to be a PhD student in my lab, and now she is an independent researcher. She's a faculty at Northeastern, as well. And we're very fortunate that she continues to lead this effort together with multiple other people. Just in case you are not working with mass spectrometry imaging regularly, here is what's going on, right? So if we have a slice of a biological sample, such as tissue, here is a mouse brain, what we can do, we can consider a location on the tissue and acquire a mass spectrum corresponding to this location, and then we can take another location, acquire another mass spectrum, and take another location and acquire a mass spectrum. And so this way we can systematically, raster or sample different locations in the tissue, which helps us understand the molecular composition of different regions. And the type of molecules we're talking about depends on, many aspects of the workflow, but this can be peptides, this can be proteins, this can be lipids, this can be other types of small molecules. And so, as you can imagine, this type of experiments generate data that are quite complex, especially if we consider not just one tissue section, but maybe we can have serial sections, let's say, of the same mouse. So we have different slices of the tissue, which are similar because they're from the same animal, but different because they're not identical. But then maybe also we have different mice. And so now the mice have some mouse-to-mouse variation, and they're not quite the same. But then maybe we have some groups of mice who are wild type, and then another group of mice who have a knockout or are treated with a drug. And so now the question is how the chemical composition changes spatially within a tissue, but also across the tissues while accounting for all the complex types of variation. And so if we think of it like that, so there are really different questions we can ask depending on which type of direction in this data structure we are looking. So for example, what we can do, we can just consider individual spectra. And there's a lot of analytical tasks that take place there. By the way, it's part of the difficulty with mass spectrometry imaging that we don't have a chromatography step, right? We don't have a separation, and so this creates additional challenges. And so in particular, we may want to work on deconvoluting overlapping signals, looking for isotopic distributions that overlap, define multiple peaks that are fragments of the same analyte or somehow belong together without the ID. Obviously, annotation of the spectra is a really big challenge. And we also want to work with more complex data, such as ion mobility or other types of additional dimensions, which allow us to clean up a little bit some of these signals. And of course, eventually we want to work with multiple measurement modalities, maybe different charges, but also maybe not necessarily with mass spectrometry, but looking at other types of modalities as well. So this is what happens if we look at this just from the kind of spectral perspective. On the other hand, what we can also do, we can focus on a particular M over Z, so a particular analyte, and look at the distribution of abundances of this analyte across the entire tissue. So this would give us the single ion images like that. And so if we think in terms of the analytical problems, from kind of coming through the statistical mindset, I like to think of these problems as three kinds or three groups, all having something to do with the classes of analytes, right, or classes of molecules, or at the same time classes of samples potentially, right, or classes of locations in the sample. For example, class discovery means that we would want to look at the locations in the tissue which are homogeneous with respect to their chemical composition. So class discovery says, okay, let me find some regions in the tissue which have similar distribution of one analyte or maybe multiple analytes. This is relevant because maybe these regions share some morphology, maybe they share some disease characteristics such as tumor and so on. But maybe also there's animals that are treated with a drug, and you want to see which regions of the tissue, which regions could be accessed by the drug. And also, even like mundane things such as, okay, what is tissue and what is the background? Sometimes it's actually not clear. So this is for the imaging purposes, we can think of segmentation. But in the statistical language, it says discover subsets of the pixels, subsets of the regions, which are similar in some sense. Then the second is class prediction. And so this is different now because instead of just saying in an unsupervised manner, I find something similar, it is a supervised approach where we say, okay, I have some regions in the tissue that are tumor and others are healthy. I want to be able to predict in the new tissues what is a tumor. Or maybe I want to predict, some other characteristics. And so this is different, and I distinguish this because it would require different statistical methods and different kind of tools. And then the last one is class comparison, which is, again, a different question. It would require different statistical methods. It is, we want to know which analytes change in abundance between conditions across multiple animals, for example. And so this is also quite complicated because it may not make sense to average the entire tissue. You want to look for things in different subregions. Different pixels are correlated when they come from similar spatial locations. So how can we develop statistical methods that account for that? And of course, kind of thinking a little bit beyond mass spectrometry, what if we have additional modalities which are not just mass spectrometry, how we work with that? So in other words, there's work to do on the spectral side of things. There's work to do on interpreting quantitatively on the tissue side of things. And of course, as a computational person, there's quite a bit of work to do on the software implementation side of things, because increasingly the files are large. We're talking like gigabytes and gigabytes of data. So we need to make sure that the tools can handle this data, right? They can scale, and not just to the size of the file, but also to the complexity of the experiments in terms of, if you have multiple replicates, multiple tissue sections, multiple annotations, you need to have an infrastructure that keeps track of all of this complexity so that you can subset things or account for things. So this has to really consider the entire experiment as opposed to one spectrum or one tissue. It has to be accessible to scientists who do not necessarily are AI, machine learning, statistics experts. And we feel very strongly about open source tools because it allows us to, do reproducible research, but it also allows other people to question our methods and say, okay, well, we don't like how you do it, we can do it better. Well, that's good for science, right? And in addition to open access to tools, what I also feel very strongly about is open access to data, because it is often not clear how a particular method came to be if you don't have the data that actually support that. And so we need to think of this as the entire ecosystem. And so I just want to preview, because as luck would have it, next week, the International Mass Spectrometry Imaging Society Conference is taking place here at UC Davis, starting, I think, Sunday or Monday. So very soon. And then just, great timing. So it allows me to preview. Essentially, I have 4 of my lab members, Kylie, and 3 PhD students in the lab. They will be here with posters, with a lot more details about work. So if anything that I talk about today resonates, make sure you catch them at the conference, because this is, they're really happy to talk about what they do. Okay, so now in terms of the overview of what Cardinal is actually doing. So Cardinal, when Kylie did her PhD, she does a PhD in statistics, so she thought of doing kind of some, statistics and mass spectrometry imaging. And very quickly she realized that before she can do any statistics, she has to work with the data. She has to get the data into a form that is amenable to statistical analysis. And so, therefore, a lot of her work really focused on different steps that prepare the data for statistical modeling. And so in particular, in terms of import, so our default format is IMZML, and this is an format that you can have like converters from proprietary formats into that. But we also take text files. So if you have a text file which has XY coordinates and some intensities, this could work. Visualization. So before we kind of do much work with that, we want to look at single ion images. And there are quite a few techniques there in terms of contrast enhancement, image smoothing, scaling, choice of colors, a lot of kind of things that go into that. And this is on the single ion kind of side of things. But then in terms of the mass spectrum, we may want to look at individual spectra, overlay multiple spectra, look at the mean spectra, and kind of look at some basic statistics and so on. And then the next step kind of goes into this direction of, well, we actually need to figure out what is meaningful for us to analyze. In particular, maybe instead of the entire tissue, we're interested in ROIs, so regions of interest. Or maybe some of the spectra, maybe we added standards to our samples and we know where the standards are, we can add some annotations. Also, it is useful to do filtering. So for example, we can filter, M over Z ranges. We can filter different spectrum pixels and intensities. Combining samples, this is for multi-tissue experiments, this is really important. And then, basic housekeeping like log transformation, yes or no, what happens to missing values, what happens if after all of the kind of, initial steps, you end up with some negative, intensity, sometimes it happens, and so on. Okay, and this is really important, right? And then the pre-processing, you see it's the longest series of steps, because this is really what needs to be done before we can proceed with some kind of analysis. And this is the longest list. And in our experience, this actually is very impactful, right? So, statistics and AI is very fancy, right? And glamorous, right? Well, this is the basics, right? Because if we don't do this step right, all the downstream work can go wrong. In particular, normalizing with total ion current, and other options, baseline removing, median and others, smoothing of the spectra, there's many options, spectral alignment, how we align the spectra across pixels, and especially if we have different samples and different tissues, recalibration, peak detection, peak alignment, peak integration, so all of those things that need to be done, so that now we say, okay, here's an M over Z, here are the intensities across all the pixels, across all the samples. Let's see what changes, right? Or something like that. We need to actually prepare this information. And now, this is the part where, it becomes very interesting for a PhD student in computer science, for example, where we can say, okay, how we can work with class comparison, class prediction, class discovery in ways that are meaningful for this type of spatial experiments. And this is the part I will focus on today. And then, of course, there's also the export part, how we can export the data, the image files, and the code and all of that to ensure reproducible research. Now, all of this is done in R. At least our users think that they're doing R. So in the sense that when you interact with the data in this way, you are writing the R script. What actually happens in reality, that it is all C++ implementation. Not all, but like a really big part of it, especially for the very first steps, which deal with sparse data. So it has to have some very efficient data structures that are scalable. Many of our users, they don't have large computing infrastructure, so they have data that do not fit into memory. So we also support out-of-memory computation, parallel computation, and things like that. And so there's actually quite a bit of work led by Kylie that is done on this kind of backend side. And as a preview, so you will not hear about that next week, but we are working on also providing Python bindings in addition to our bindings for that. So hopefully it will be even more multilingual and more versatile. Okay, and so now let's maybe kind of think a little bit about this statistics and AI part of thing. And so I would like to preview a little bit what we used to do some years ago and how our current thinking kind of evolves from that. And I would literally go through the three parts: class discovery, class prediction, and class comparison. So the older method this Kylie developed as part of her PhD was to say, okay, well, I'm interested in finding regions of homogeneous chemical composition in the tissue. And this was a collaboration with Graham Cooks when we were back at Purdue. So this is a pig fetus. So clearly there is a complex morphology. So what you can do, you can treat each pixel as a vector of all the M over Z after they are aligned, picked, and so on. And so if you do some basic thing like k-means, for example, well, now the colors show regions where these vectors of M over Z are similar to each other. And you can kind of see the liver and the brain and some things on the spine. Now you can do dimension reduction with principal component analysis, so maybe it's a little bit better. But fundamentally, there are two problems with this approach. One is that k-means or other versions of that, they think of each pixel as an independent vector of many dimensions, right? And so they do not recognize that we actually have an image which has spatial structure in it, right? And neighboring like locations should be more similar to each other. And the other is, it is based on all the ions, right? So essentially, these regions are defined on, how many hundreds or thousands of M over Z you have. So they all define these regions, and we don't really gain much insight. And so what we did, we kind of proposed an alternative that addresses these two things. So one is we incorporated spatial information. And we did this by considering not individual pixels, but there's like a kernel smoothing around them, which essentially says we have neighborhoods. And so you see now how this cleaned up this image, because now we kind of recognize that this kind of jagged patterns, they're really artifacts of these methods. And so now we can see much more smooth image. And also we can add to this a statistical or machine learning technique called regularization, which allows us to eliminate ions which do not contribute to any of the segments substantially. And so now it would look like that. So for example, here we will have for the brain specifically, this is all the M over Z, and we can quantify the signal to noise ratio of different M over Z as related to the brain. And so some regions here, we don't see them because we say that these regions of M over Z do not contribute to the brain, like as compared to like other regions of the tissues. But others, like for example, these M over Z, they're very brain specific. And we can kind of plot it also on the signal to noise ratio, and it allows us to see what happens. And a side effect of this, by the way, is also selecting the number of segments. In k-means, K stands for the number of components or clusters. You have to specify it in advance. This regularization allows us to choose how many segments we want. So this is very useful, but as you see, this is from 2016. So that's been like a while ago when Kylie was a PhD student. And so now we've progressed more on that. And so this is the work by Dan, who graduated a couple of years ago. And so what she considered, like the thought process for her was, even though we added some spatial structure and we added some regularization, still each segment here is defined by all the hundreds or thousands of ions. So this assumes essentially that every single ion has the same spatial distribution, so that every single ion or a subset of them, they have some distribution in liver and some distribution in the brain and so on, or the heart. While in reality, of course, it is not true, right? So different ions have very different localizations. So using all the ions to project them to single set of segments is maybe not biologically meaningful. In other words, different ions have different segments. And so what she decided to do is to try a similar type of segmentation, but now univariate, where we segment one ion at a time. One ion has its segmentation, another ion has a completely different segmentation. And so the inspiration for this was also a collaboration with Graham Cooks back in the days. This is an actual oil painting. One of the students in Graham's lab painted the image. That's like a typical Indiana, farmhouse. And this is oil, meaning different colors have different chemical composition. And so now we can image this, in this case with DAISY imaging. So this would be the actual mass spectrometry image of that. We kind of match the colors so that it looks like the painting. And this will be this type of segmentation using the multivariate vector of ions. And so this is nice. And well, you see, it more or less got the houses, right? The tree. Well, didn't get the donkey here. The donkey got lost. But in general, you see the bulk of the information. But then if you look at the individual ions, you see, okay, here's a particular M over Z. It clearly is matching the black outlines of all the objects, right? You really see these outlines. And then this M over Z clearly catches this light green of the grass. And so different M over Z, so black localizes very differently than green. And so can we now actually segment this M over Z separately and this M over Z separately so that we can now automatically say, okay, this is the collection of pixels that corresponds to color black, and this is the collection of pixels corresponds to color green. And that's what Dan ended up implementing, and this is actually a very useful technique that we use now. But then Dan kind of kept thinking further. Okay, so it is very nice to be able to have separate segmentation for different analytes, but obviously when we have only one ion, it can be very noisy. So the advantage of this multivariate segmentation is that when we project all the ions into a single set of spectra, the information accumulates, right, or compensates for each other, and so it's much cleaner. So for a single ion, we have to overcome quite a bit of noise. And so she asked herself if there is something in between. Maybe we can have segmentation which are not all the ions together and not just one ion at a time, but maybe there's some kind of gold middle ground where a subset of ions give us the right segmentation for different localization. And so this is a simulation that she did to motivate that. So there are orange color is some kind of background, and now she has two circles. So the gray circle is a smaller one, and then the blue is a larger circle on top. So for some ions, you have only the background. For others, you have only the gray circle. And for other ions, you have a large circle. And so this is what a simulated single ion image looks like for cluster 1, and this is for cluster 2. And so if she now tries to aggregate, find a subset of clusters which belong together and aggregate them, so because it's simulation, she knows the exact answer, right? So if she just does one ion at a time, the accuracy is so-so because of the noise. Now she aggregates multiple ions that should belong together. The segmentation accuracy increases. And then when she has kind of even more ions, then she starts averaging things that do not belong together, and the accuracy again goes down. And so she came up with this method that does that. So this is one of the data sets that we have. This is what all ion segmentation would look like. This is what a single ion segmentation for one ion would look like. And you see that this ion has a fairly different spatial structure from this. And now this is ion clustering that combining all the ions that have a similar spatial localization. And so now you see that this kind of structure becomes much more pronounced. And so the subset of clusters, they may be adducts, they may be fragments of the same molecule, they may be closely related in some kind of pathway. We don't know that because we don't have the annotation, although, for example, for isotopes, we can kind of see the mass difference. But this allows us to do two things. One is to clean up the signal, and the other is reduce the total number of features. And this by itself is also interesting because the high dimensionality, the more artifacts we can have. So this is what she was doing for segmentation. Now, this was the unsupervised method. Now, the second is supervised. Remember, we talked about class prediction. So the context for class prediction is oftentimes in a clinical setting where we want to predict regions of the tissue which are specific to tumor, for example. But sometimes there are other, or like drug localization or something like that. And so the old methods, I'm not going to talk about them, partial least squares, maybe logistic regression, there's like all the kind of traditional classifiers that you can think of, right? And so one of the projects in during Dan's PhD was to say, well, let us consider issues that can arise specifically in the context of mass spectrometry imaging. And so here is the issue that she kind of identified in particular. So if we want to train a classifier that predicts if this region of the tissue is tumor and this region of the tissue is healthy, we need to have some examples, right? Because we don't know otherwise. So we need some training data, and the training data typically comes from some kind of annotation. And annotation would be like in the, for example, oncology context will be some pathologist, right, who would say, okay, there is a tumor here. Now, here's the problem. Pathologists do not have a spatial resolution of a mass spectrometer. So typically what they would do, they would draw a circle and say there's tumor here, right? They will not go like pixel by pixel and say this is tumor, this is, and by the way, even if they do, they may be wrong. But generally they would say, okay, here is a region with tumor and here is region with no tumor, which means that in reality, our training data, which are marked as tumor, in reality are a combination of a tumor and non-tumor tissues. So healthy biopsy, typically we can think of this as healthy because we know, it's taken from a different region and so on. But what we think is an example of tumor is actually a mix of a tumor and non-tumor, most often. And so for classifiers now, if we think that this is tumor and this is non-tumor, the classifier will be biased because in reality, our training is labeled incorrectly, right? And so what you can do instead is to acknowledge that and say, okay, I can think of non-tumor tissue as being pure and clean, but now that what is labeled by the pathologist as tumor is actually a mix of tumor and non-tumor tissues. And so this is a kind of very common setting for semi-supervised learning, or also called sometimes multiple instance learning, where essentially what you do, you look for pixels in what is labeled as tumor that are similar to healthy, and you relabel them as healthy. And then if they become different enough from the intensity or abundance perspective, then you say, okay, they're different, therefore they're tumor. And so it turns out that this really helps a lot in terms of classification accuracy. And so this is also something that she implemented as part of Cardinal. So this was an example with some tumor biopsy cores. And we were actually lucky because not only we had the training and validation set, but our clinical collaborators, they painstakingly actually went and labeled every pixel just for the purpose of this collaboration to also manually verify if this prediction is improved, and it really is improved quite a bit. Okay, so this is on the, so we just talked about segmentation, so class discovery, and class comparison, classification, right? And so we just talked about the fact that segmentation would involve fairly different methods than classification. And so the last part, class comparison, I find that this is probably the most underdeveloped in a way in mass spectrometry imaging. And I would think it is because unlike the previous methods, which can be done on one tissue or like a small number of tissues, if you want to know which analytes change in chemical composition between different groups of individuals, well, you need to have groups of individuals, right? We need to have examples of samples from different groups and different conditions. And increasingly, though, we can have this type of data because the throughput of the experiments increases. We have kind of more access to this. And so this is the work led by the PhD student who is now in the lab, Ethan Rogers. And so I would like to kind of take a little bit deeper dive for that. And by the way, so I mentioned in the beginning that we do a lot of proteomics work, and we kind of do some statistical methods for proteomics. The work that Ethan does for MSI is very much inspired by our experience from kind of similar problems in proteomics. But before I do that, maybe if there are any questions that we can kind
of. Mine will still be when there's margins that we know the signal fluctuate per pixel. We kind of smooth them or use the neighboring effect. But when it comes to now it's like a single cell coherence, how do we know it's artifact versus we're missing something unique that's actually true signal?
Yeah, you don't, so the way these methods work, you don't just smooth, you don't just dilute the signals, but you say, here's a neighborhood, but we allow for the possibility of the signals between neighbors to be quite different. Imagine, for example, you're on a boundary of some morphology, right? So you will take the neighborhood, but you would allow for the, so your prior information says neighbors should be similar, but you allow the data to overrule that. So essentially that's what works.
And we also came from like untargeted omics and then moved to imaging. And there's, I think 15 years ago, the QC becomes more important and more demanding to have QC. Whereas on imaging, it's more about, yeah, I look at the painting and then it looks correct. Or when you compare different tissues, it's like, let's normalize to total and they should pop up. It's more like a feel, or it takes a lot of experience to judge that. What would be your thought or consideration to move to a more quantified QC in imaging comparing multiple samples?
Oh, I love this question. You know, there are many things that can be done, including indeed building QC in the experiment, like adding standards and things like that. So for method development folks, you know, this oil painting is amazing, right? Because we look at it, we know what to expect, we understand that the method works, right? For more biological experiments, adding standards, working with systems where we understand some biology and we have some expectation. And, you know, I would do it step by step because the problem is more complicated altogether, right? And so probably what I would also do is to break the computational problem into different steps and make sure that we have QC for different steps and we can try to understand where the problems come from primarily. And some of the steps can maybe addressed maybe in a general way, like using computational tools or experimental design. And others would require, you know, specific standards, right, for different parts of the workflow. And then we would need to have analysis methods that incorporate that. So maybe as we go into the deep dive here, maybe it will kind of clarify. But long story short, there's a lot to do. That is for sure, right? So I'm not at all saying that this is all easy and solved. And yeah.
You see some groups, like or dozens of groups or hundreds of groups are already pushing it to that work. The QC on the technical part, say it's not in-source fragment, or QC on the data massaging part, and we're not massaging too much. I've seen one or two papers on talk about fair data on imaging, but that's pretty much it. But you must know more than I.
You know, there's a lot to do. And, you know, I would say as a computation, so we're a dry lab, right? We don't have a wet lab at all. And so for us, access to data is a really big deal. And so since I'm working kind of in proteomics and in imaging, I would say that it's much easier to find data in proteomics than it is to find data in imaging, that, multi-lab comparisons or different QC approaches in a way that we can access them and we can work with them. Maybe we can kind of talk a little bit more after that.
How granular does the pixelation get on these images? Like, to some extent, a pixel is going to be like a region of space in the tissue you're looking at. And so I guess, can the pixels get fine enough to be more or less on an individual cell basis, or is it?
That's what people are working towards. Not always, right? And not the data that we have here, but increasingly this is the interest. And people like, I don't know, Theodor Alexandrov or others, they're getting, so in fact, next week you will hear a lot about them. If you come to the conference, so essentially, like trying to kind of go really deep to spatial resolution, right? So absolutely. So here it is not single cell level, the data I'm showing, but there's a lot of interest in some labs are getting close to that.
I don't want to hold you up any longer, but we can discuss later. I had just a question about the validity of the classifier concept in today's time in general, but we can discuss that.
Okay, let's talk about that after that. Yeah.
What kind of algorithm did you use to move from k-means and PCA towards including the spatial information in the neighborhoods?
What kind of argument?
What kind of algorithm?
Oh, algorithm. We came up with that. So essentially, instead of a single method, you have a kernel that has like a smoothing around that. And so you expand the vector. So very intuitively what happens, so you have a vector of M over Z for one location. And so let's say you have a neighborhood of 9 pixels. So now you concatenate 9 times M over Z of all the pixels, except that the pixels which are further away from your pixel of interest, you downweigh them. So that's kind of a very intuitive way, but it's published, so you can take a look at the paper. Okay, let me do a little bit deeper dive in kind of differential abundance, because this is on one hand understudied, I believe, but on the other hand comes very much from our experience that we have in proteomics. So for class comparison, I'll just give you a example. So this is a collaboration with Peggy Angel and Birgit Schilling. I know Peggy will be here next week for sure. Birgit, I'm not sure, but please come and talk to these folks because that's really great work. So this is an example of bone imaging, and bone imaging is actually yet another level of difficulty experimentally, right? And so the idea is to understand how the bone and the cartilage, how they get affected by osteoarthritis, right? Osteoarthritis is bone damage, right? So the cartilage kind of gets eliminated as the bones kind of, touch each other. And so it was interesting to see how this affects the molecular composition. So this is the workflow that we think about in terms of looking for changes between, for example, healthy and disease joints. And the first step, like if you know me, you probably know that experimental design is like the key thing that we need to worry about. So this was a very nice experimental design. So they had subjects with osteoarthritis and subjects which were controlled, so they did not have osteoarthritis. So these were cadaveric samples. And they had 4 individuals from each group. And what is interesting, they also, for each subject, they took 2 paired samples, medial and lateral, so both sides of the knee. And so this is what the single ion images would look like. So we had 16 images, half of the subjects from osteoarthritis, roughly 45K unique M over Z in this M over Z range. So there you go. So now we can kind of talk about data processing. So what we do. So there are multiple steps, in particular mass calibration. So align spectra across samples and pixels. There was a standard, kind of to your question. The standard really helped in that regard. Then peak picking. So we take the peaks and we kind of try to identify the peaks across multiple M over Z. And so this allowed us to reduce quite a bit of M over Z signals to a relatively small number of features. And then normalization also kind of, there's not a single well-accepted way to normalize the data. So we actually came up with our own custom normalization. So it's kind of like a median normalization where we equalize the median values. But in MSI, we found that there were a few outliers that really affect even the median values. And so we had to come up with some truncated version of this normalization. By the way, if you're interested in that, Ethan, who will be here next week, he will actually have the actual code on his poster. So you can see exactly how it is implemented. We're not hiding anything. And so now the interesting part here, because we have bone and cartilage and like other things too, is the definition of regions of interest. So the actual ROI. And I would like to take a minute to talk about that because this is something that is specific to imaging that we did not have to worry about in proteomics. It is because we have the spatial structure. So this is a simulation, and we like simulations because they help us understand what is going on. So Ethan designed an experiment with two conditions, A and B. And these are our tissues, they're rectangles. And our regions of interest are these donuts. So this is the region that we're interested in, and like outside, this is just background or something else. Now, for this region of interest, he simulated some noise features, just randomly with some spatial correlation, which have nothing to do with any condition. We just add them there. And then there are some features that are differentially abundant. So within this donut, condition A has something different going on than condition B. And so the question is, can we find within this region of interest a subset of analytes which actually change in abundance between A and B, while setting aside the analytes which do not change in abundance? And so this depends on whether we know what is the ROI. So if we have the information about how we define the region of interest. So for example, if I tell the algorithm this is the feature that defines the ROI, well, I can very well find the ROI, right? So there's no problem. But oftentimes we don't know. We just say, hey, here's cartilage. Let's segment the tissue to figure out where cartilage is. How we segment? Some multivariate method like k-means or spatial shrunken centroids or something like that. So here's spatial shrunken centroids. This is how it attempts to segment this known region of interest. Clearly, it is not doing a very good job, right? Why? Because we have all these noisy features which compromise that, right? And the method doesn't know which features are useful, which features are not useful. It just looks at all the signals and says, okay, these are the segments. K-means does similar. So it's really not a problem of the algorithm, it's a problem of the structure of the data and how much noise we have. And by the way, the more dimensions we have, the more these artifacts will come up. And so now what people do, and by the way, this is also something that people do in single cell genomics or transcriptomics, for example. They will say, okay, I found a segment in condition A, I found a segment in condition B. Let me see if there's a differential abundance between the segments in A and B. My point is that this is a really bad idea. And so this simulation shows why. So first of all, let's take a look at which features actually define these segments. So here's for spatial shrunken centroids. So this is the true differentially abundant features because we know what they are. This is the features that define segments in A, features that define segments in B. You see that the overlap is 4 features. It's really not related at all because of the artifacts of the noise. Same thing, spatial k-means, not much overlap. And so now if we try to compare this segment to this segment, well, if the segments are defined by the features which are known, we find these differences very well, right? But if now we say, okay, let me compare this segment to this segment, we don't find anything. Why? Because the segmentation methods already absorbed all the differences in the signals, and there's nothing else left to compare. Now, this is the case in this particular simulation. Sometimes you can have artifacts, you will find some kind of differences, but again, these were all affected. They will be all affected by the artifacts of random variation. So the message of the simulation is it is really not a good idea to use the same data twice, once to find the ROI and then test for differential abundance. And in statistical language, it is called data leakage or double dipping, where we use the data tautologically, once to find the segments and then to compare them, right? So that's not right. And so instead, what we need to do is something like this, where we have some features which we would use as markers for particular regions of interest. Hopefully these features come from the literature or from some kind of other external information. We use this for segmentation, set these features aside, and then we start comparing all the remaining features. And so that's what we try to do. So there are some markers in the literature for cartilage and bone. And so The bone markers were kind of tricky. So we first segmented the cartilage, and then within bone, we could segment again, but only with respect to those features. So not the whole data set, but only the features which are to be known markers. And so that's kind of what it looks like. You see that the cartilage really stands out from backgrounded bone. So this was useful. And then background, you kind of can segment this a little bit more with this marker. And this marker was not super useful in our data set. But it allowed us to use external markers to find the regions of interest, which we can compare in terms of differential abundance. Okay, and then feature filtering and feature aggregation. So now that we have regions of interest and we already have picked features, we can come back to the same idea that Dan had before, that if we combine multiple features which are spatially redundant and aggregate them, we will reduce the number of features that we test. And in statistical language, it means we reduce multiple testing, so it gives us more sensitivity. And that's what we did. So this is just kind of documenting what happened with this data set. So we started with 45,000 M over Z values. After picking, we got it down to 511. After we removed features used for ROI, 503. And then non-specific filtering. Non-specific filtering means that it is not specific to conditions A or B, such as, the feature has no variation at all across any pixel. So it doesn't really do anything. Or it's only present in a very small percentage of pixels and it's NA everywhere else. So some, ideas which are not really informative of the conditions, so we could remove it a little bit more. And then we can look for features which have very similar spatial profiles. We can use the method that Dan developed. Ethan went with another method called deep ion because it's more specifically incorporates the differences between isotopes. But this is what it would look like. So for example, here is one particular group of ions, right? And so like here is two that behave very similarly, right? Here is another two that behave similarly, and here's like triples. And so he decided to aggregate them by taking the ion with the highest intensity. But you can imagine taking the mean, median or something like that. And so all of that ended up with 366 features as opposed to, 45,000 in the beginning. And so now statistical modeling, and I'll just kind of go very quickly. So another simulation, which tries to help us understand what kind of statistical models are useful in the context of imaging. And so the simulation tried to mimic our osteoarthritis data set. So here we have measurements with, again, blue are osteoarthritis, pink are healthy. So here we have a simulation with a lot of technical noise. And here we have a simulation where main driver of variation is person-to-person variation. And so what we can do, we can compare now healthy versus disease, so pink versus blue, or within each person, we can compare medial versus lateral. So one is between people comparisons and the other is within people comparison. And so now it is fairly easy to see that, especially in experiments when the person-to-person variation is a big driver, the within subject comparison is much more sensitive and it detects a lot more things. Why? And this is very kind of typical thing in statistics, because within subject comparison, each subject serves as their own control. So you kind of eliminate the person-to-person difference by comparing the medial versus lateral of the same subject. And you can do it using this family of models called mixed effects models, where you incorporate all the sources of variation. Now, when the biological variation is smaller compared just to technical noise, it doesn't really get you very much. But when the biological variation is the main driver, meaning the technical variation is well controlled, this accounting for these different sources of variation is beneficial. And this is as compared to, taking pairs of conditions and doing a t-test between them. So not modeling the entire experiment, but just looking at pairs of conditions, you see that you gain more sensitivity by modeling all the data together. So here the green is the true positives and the orange is the false positives. It's a simulation, so we know exactly what's going on. So this is one message, and the other message, and I see this a lot in mass spectrometry imaging, where instead of doing something like that, what people do, they say, hey, but we have a lot of pixels. Let me do a t-test, and now each pixel will be a replicate. Now, clearly all the pixels, they come from the same person and they're spatially similar, but your t-test will think that there is a lot of replicates, right? Because we have many pixels. And here's another simulation which repeats the same thing, but now we analyze this thinking that our pixels are replicates and we see how bad it is in terms of false positives. It will be very sensitive. Almost everything is significant, but just the same, a lot of false positives, because your t-test thinks that you have thousands of replicates. Of course, everything will be significant. So this is something to avoid. And of course, what we can now do is to take the actual biological data, understand how much variation we have. And these mixed effects models can be used to plan future studies. In particular, we can understand how many replicates we would actually need to find differences between the groups if we were interested within subject comparisons versus between subject comparisons and so on. And we can also play out different scenarios. What if we expect only a small percentage of features changing versus a large percentage of features changing? And by the way, this is also a classical fact in statistics that if we have an experiment where only a few things changing, we need a larger sample size. And this is because we try to protect ourselves against false positives. And if we have a very small number of changes, of true changes, it means that we have a lot more opportunity for false positives. And if we have more potential false positives, we need to work harder to protect ourselves against that. And work harder means larger sample size. So that's how it translates here in imaging. Okay, and so now I have 7 minutes left, but I will probably not even take this 7 minutes to tell you about the very last part. Also, I will be here next week. So the big message of the previous case study was that we need to have ROIs externally determined. So not reuse the same data to build the ROIs and then compare them. So externally determined how, right? So ideally, we would have some M over Z that we know are markers of something and we just measure them very nicely and they have nice intensity. We can outline the ROI and all is good. It's rarely the case, right? We may not know what the markers are, or maybe if we know, we just are not able to measure them accurately, or they have interferences or something like that. So it is really useful to be able to define ROIs using some kind of other external information. For example, using other modalities than mass spectrometry imaging. And for that, what we need to do is to lay over something like H&E experiment, right? Or maybe, I don't know, spatial transcriptomic experiment, right? Or something else. And use these modalities to determine regions of interest and then kind of, carry over this region of interest to MSI and then start the comparison. This will not do any double dipping because we don't use the spectra twice, but we need to be able to transport the annotations from one modality to another. And this is actually very difficult because images acquired by different modalities, they can be deformed. And the deformations can be due to many sources, tissue handling, but also multiple imaging runs on the same tissue, transform the tissue. Sometimes you cannot do multiple modalities on the same tissue. You have to have adjacent sections, which are not identical. And so you try to essentially transport the annotation from one tissue to another, and the grid of the measurement is determined in very kind of flexible and elastic ways. And so this co-registration is actually very tricky because, for example, if we think of MSI and H&E, the nature of the data are very different. H&E has 3 channels, RGB, right? And MSI, tens of thousands. Range of intensities are very different. Spatial dimensions, so H&E has much higher spatial resolution. Different information content, right? So MSI is typically much more localized for different ions. So this is to say that we cannot just take some kind of off-the-shelf method to co-register things. We really need methods that are dedicated to this type of data. And methods exist, right? But many of them have limitations. So for example, many methods, in particular in Galaxy, they require some fiducial markers. So somebody has to annotate the tissue with some markers that can be aligned, right? And all the methods typically support affine transformation, so, right, not elastic transformation. Now, more recent methods actually try to view this as an optimization, and they optimize some kind of more complex loss function. However, typically they need to minimize this with respect to some reference. So we need to figure what is the reference image to what everything is co-registered. And then the more recent kind of approach will be using some kind of neural networks. But if you're not careful, neural networks will require a large training data sets. And we may not necessarily have that. And so Sai kind of did that, but because he spent so much time working with mass spectrometry imaging, he tried to really adapt the architecture of neural network to the properties of MSI data. And the specific thing, well, there's some issues such as processing and pre-training, which are very specific to MSI, but the biggest thing is the attention maps, where, remember, different ions have different localization. So neural network will focus on the attention on different regions of the tissue for different ions and use this to guide the co-registration. So we don't just use the full vectors of measurements for every pixel, but we just use subsets of measurements. And this really helped quite a bit. And so here are some examples. Sai will be here, please talk to him. So here is one example, H&E versus MSI. This is what happens before co-registration. So that's how it happens after. Note that there is no fiducial marks, no manual annotations, elastic transformations, it all kind of works. And now if we have more complex things such as cores, we can co-register them, each core separately in the same image. So this is also quite useful. And now more recently, we actually are working on more kind of broader co-registration with other modalities. So here, for example, we have, so this is a data set from Livia Eberlin's lab. So this is DAISY MSI, which has post-MSI H&E, and then Visium spatial transcriptomics, and also Visium H&E, which is kind of comes together with spatial transcriptomics. And so I am kind of not going to go in the technical details, but fundamentally now that this type of approach allows us to work even beyond MSI and H&E to work also with transcriptomic data sets and have 3-4 types of modalities co-registered together. And Sai came up with different metrics which characterize how well this co-registration takes place. So he will be happy to tell you more next week. So let me just wrap up. So in terms of the recommendations, I think increasing the signal is a good idea. So define ROIs, which are exogenous and possibly from co-registration. Use subjects as their own control. So paired designs are really good tools that allow us to eliminate some of the biological variation, and we should use that. Increase sample size, right? That is obviously the case, but we do have tools now how we can learn from past studies to plan the sample size. So it's not like we need, as many replicates as we can, but we can plan it out here. Reduce noise, use QC standards and normalization, right? So there's quite a bit of work that needs to be done. Address sources of missing values such as outliers and zeros. We will be working on that for a while, I'm sure. Reduce the number of comparisons by aggregation of features and things like that. Use appropriate statistical models. Do not use pixels as replicates. Describe all sources of variation. Verify model assumptions. Correct for multiple testing. Now, if none of that makes sense, or it's really overwhelming, or it's just not sure where to start, then I only have one very simple message to you. Talk to a statistician before collecting the data, and talk early enough so that we can work together, and both on the experimental design stage and also on the appropriate analysis stage. And speaking of talking to a statistician, I would really like to take a minute to advertise the educational effort that our lab has been leading for the last 11 years. So we lead a program called May Institute, May like in the month of May. So it actually happens every year in May in Boston on the campus of Northeastern. So this is a 2-week, you can think of it as summer school, except that you don't have to be there for the full 2 weeks. It's actually programs which are, 2 days, 2.5 days, or even 1 day, and you can pick and choose any subset of programs that are useful. So a lot of this, or quite a bit of this, is focused on proteomics. So we have proteomics with Skyline. This year we also have FragPipe coming for us. We do basic R, basic Python, and basic statistics. I actually teach quite a bit of just basic statistics lectures. We do a lot of work on statistics for proteomics and what are the methods and how it works. And we have a module on mass spectrometry imaging. This year, we're going to double the time for mass spectrometry imaging just because there is so much demand. And the way we double it is not just like more lectures, but we will actually give more space for bring your own data and bring your own case studies so that we can actually sit down together. So we really mean it, right? We can sit down and kind of think about, okay, what are the formats? What are the data structures? What are the methods that fit well? And all of that. And I also mentioned at the beginning that we have another interest in the lab is downstream interpretation of analytes in the context of biology. So our colleague, Ben Guri, he develops a knowledge base based on natural language processing, which mines, literature, PubMed, etc., and creates a structured knowledge base, which is really exceptional. And so now our proteomics work interacts with this knowledge base directly. And so there's an opportunity to learn about that, how we can work, how we can go beyond p-values essentially and understand what happens on the biology side of things. So this is in person, and our audience is a combination of students and people from industry. And in the past, it has always been like a really nice driver for community interactions. And so if you can, please join us. If you can't, maybe pass on the message to your friends and colleagues. I hope to see you there. And now the most important slide for me, I'm here talking, but the people who really do the work, there are many of them. And Kylie, I haven't had a chance to mention Melanie, Dan, Sign, Ethan. They were really driving the imaging part, but we have a lot more collaborators who work more on the proteomics and biological interpretation side. Thank you so much.
Thank you so much. It's a fantastic overview. First, do we have questions already online? People who would raise their hand. So people who are online, please don't be shy. You will be heard. You can raise your hand, and then Diego will call you out. Then we have questions in the room. We have questions in the room over there.
Will you accept kind of a more general data science question?
Let's try. I don't know if I have the answer, but we can.
I guess just in general, how much can you process data and still have it be reliable? Because the way I perceive it is like the raw data is kind of the closest you get to like just unfiltered reality of what you found, and like each process of refinement could almost be like its own assumption almost. So that how far can you process?
I love it. That's very right. And this is how I started as a PhD student. So, or I guess when I kind of started my first exposure to mass spectrometry. So a lot of data processing is pretty much like a recipe, right? So let me subtract that, divide by this, right? And then multiply by something else, and then voila, right? Now we have processed data set, and hopefully it makes sense. And so I started thinking about, okay, what does it mean, right? Like when we normalize by something or subtract something or, process the data in different ways, why, right? And so like I take the log, somebody else doesn't take the log, right? Like what's the difference? Turns out that in many cases, all of these procedures can be thought of as the result of some abstraction describing the process that generated the data and of some statistical model. And so if we think of this from a statistical model perspective, then some operations on the data are more optimal than others. And so what you can do, you kind of step back and say, okay, not in terms of, okay, this is my process that does this and that, but this is my abstraction that generates the data. And this abstraction informs me what are the steps that can be done. And the steps can be, optimal in terms of, bias and variance, and like there's like some statistical concepts that you can kind of think of. And this guides your choice of processing steps. And so I think what is really important is to be very aware of like what we are assuming. I think the problem is not in the data processing, but in the fact that we just apply steps without spelling out what the assumptions are. Because if we can spell the assumptions, then we can consider if these are good assumptions or not, right? And then we'll say, oh, wait, this is not a good assumption. Let me change how I view the data generating process, and it will change your transformation. So there are actually general principles that, and sometimes, we don't know what these principles are, and that's research, right? But in many cases, that's how you can guide the, yeah.
We have here another question.
Yeah, this is kind of multiple questions bundled into one. and probably more elementary regards to the scope of proteomics. But for MSI, we don't do you guys do tandem mass spec to determine the sequence of your peptides that you obtain from like a tryptic digest? And then because like I was told that it's not common to do tandem mass spec, and then therefore there are certain scoring algorithms that need to be applied so that you can differentiate which protein a peptide, if you have two isomeric peptide fragments, which protein it came from. And then I guess to add on to that, you had mentioned earlier that it's common to use standard to do the mass calibration. And what is like kind of the accepted plus or minus tolerance for an M over C value so that you can match it to a peptide fragment?
Okay, so let me go with the first. Okay, so the big difference between MSI and proteomics is that we don't have the chromatography step, right? And so you just kind of take these tissues and you kind of directly, because otherwise you have to do it offline and it's just too time consuming. So which means that if you want to have tryptic digest, you need to do on-tissue digestion. And I'm a dry lab person, right? So I actually don't do that, but I've been told many times that this has a lot of artifacts, like the trypsin may not be equally sprayed, and then the tissue composition may affect trypsin efficiency and things like that. So it is my understanding that on-tissue digestion is still work in progress. So it is not done very commonly. And so therefore people either do intact proteins, depending on the mass range, or lipids or small molecules. Now, there's also a way to do peptide imaging, but that's the part where it's an experimental part, I'm less confident. But now, indeed, right? So for tandem mass spec, if you do intact protein, like it's a whole different ballgame, right? What I've seen people doing a lot more is to first do this type of thing, kind of trying to understand what is interesting, what kind of features we have, and then go back and do targeted MSMS to identify the analytes for certain things that are of interest, such as, differentially abundant and that. And there are variations on the workflow now, but this will be more experimental. So in terms of like routine use of that, I don't think this is done. In terms of mass resolution, you would need to ask Heidi next week. I don't know the details.
All right, we have a question online.
Yeah, there's two in one question. How can imaging data be quantitatively scored for disease versus healthy tissue samples? And could segmentation-based analysis help distinguish between these data sets in this case?
Okay, so first of all, just to clarify, right, that imaging mass spectrometry, just like any mass spectrometry, right, it is relative quantification and not absolute quantification, right? So if you want to do absolute quantification, you need to add standards, and that's also a whole separate issue, right? This being said, for changes in abundance, you do not need absolute quantification because the abundances, they're related to the signals, right? And so if the signals change, that's evidence enough that there is a change. And now what I was trying to say earlier, and let me actually go back to the very early slide, because the question was in the context of the disease, right? And I'm guessing biomarkers of disease. So I was talking about the type of problems, right? And so class discovery, so this is segmentation, where we look for segments of homogeneous chemical composition, and maybe there's tumor, and then maybe there's healthy, right? And I just want to emphasize that segmentation alone is not enough to distinguish healthy and disease, and it is especially not enough if we talk about biomarkers of disease. Let me tell you very briefly why. So a biomarker is supposed to be actionable, right? So it's something that a clinician can, look at and make a decision what to do with the patient. Now, segmentation or class comparison, differential abundance, they are all asking at the average pattern, right? Or average abundance across different regions of the tissue, right? Or across various individuals. But the doctor doesn't see an average patient. The doctor sees a specific patient. And for the doctor, it doesn't matter that on average something happens. They need to make a decision for the specific person in front of them. And because the question is not about averages, but about specific individuals, it requires different statistical methods, right? So these classifiers their objective is to classify the status of the next person. That's what we need, right? As opposed to say on average between healthy and disease, something is happening. And, come to my institute, we'll talk a lot about that, right? How different methods are appropriate for different questions. This said, focusing on regions of interest kind of externally will definitely help focus your analysis and will definitely help focus the classifier.
Banjo? Oh yeah, hi. It's a very interesting topic. I'm not very familiar with the imaging, but I learned a lot. So my question is, so for me, it sounds like the MSI question is very similar to a CT question, like CT, in CT scan, like there are a lot of doctors using AI-aided tools to help them to diagnose like a stroke. So, and CTR just like the MSS. So my question is, did you guys benchmark using like a supervised model with like something like CNN to help to diagnose and identify the biomarker? Because it sounds like it already helped you to first segment and aggregate different layers, and then it also helped you to identify the region of interest. Did you guys benchmark this?
Well, this is one of the modalities that would be kind of really interesting to look. As a matter of fact, Dan, who I was just talking about her projects, right? That's exactly her job now. When she graduated, she went to a company that combines multiple modalities, including CT scans for breast cancer diagnosis. That's exactly what kind of she went for. But like for the research part, just to keep in mind that MSI is invasive, right? So you actually need to extract the tissue to study it, and CT scanners you don't have to, right?
Yeah.
And so it kind of limits the type of studies that you can. The devils in the details as always, right? So it's probably not exactly the same, but of course, right? So there's another spatial modality, another view on the same sample. So I am sure that there is like really interesting compatibilities between them. Yeah, we haven't done anything yet, but talking to Dan, they're doing quite a bit of that, like in the new place where she works.
Shane?
Talking about multimodality, like the example you have the small metabolized imaging versus the transcription imaging, and then the first question is how to co-localize them, and how do we proceed after that? You just correlation, or how do you map them to information together?
That's something we are working on right now. But same thing, right? So you have a longer vector, right? And so each spatial location, right? So before you had the vector of M over Z, now you have a longer vector of also transcripts, right? And so then it depends on your question, right? Such as, do you want to segment with respect to a longer vector? Do you want to see what is different? But you also may want to see what is co-localizing, right? Like which transcripts co-localize, I don't know, with small molecules or something like that. And the problem is that now you have this new vector, which has very different noise properties compared to the MSI, right? And so we are just in the early stages of that. But what we're seeing now already, you cannot just take this twice as long vector and just put it all together, right? Because, for example, like some things can be more like look like counts and other look more like continuous intensities, right? Nature of missing values is different, and so there will need to be some modeling work. But the specific methods will depend on the questions, and so it's really useful to frame the biological question. A big part of what we do is to take the biological question and translate this into a statistical question. Because if the statistical question is framed well, then you know which approaches would fit, right? And so that's what we're doing now. So I don't have an answer yet, but hope we will, maybe next year.
We have another question online.
Is there any effect on ionization or loss of signal observed of small molecular weight metabolites in the presence of lipids or in heterogeneous tissues in MSI?
We haven't investigated this specifically, but I am sure. Because we do see a lot of like things like ion suppression, a lot of missing values, a lot of interferences. And so when you have things of very different range of abundance and, different chemical composition. So I don't have a very specific answer, but I'm sure that's a source of artifacts.
This is more of a clarification question, but are the literature ROIs generated from AI determining or like multiplexing your IHC data and your MSI data?
So no, in that case, it was like literally from literature. Somebody else published a paper previously, and they found this M over Z, they were specific to cartilage. So that was like literally from a paper published previously. And so the point is that you should not be using the same numeric values twice, right? The same intensities of M over Z twice. And so if your ROI comes from a different source, such as previously published paper, or measurement on the same tissues, but from a different technology, right? A different modality, that is fine because you do not use the same numbers twice. So the danger is to literally use the same numbers twice.
All right, yes, over there, please.
Do you use any external validation data to test the robustness of your models?
I would love to if we have them, you know. So as I said, we are a dry lab, so we rely on the kindness of strangers, you know. If we have experiments which have this, we love that, but it's not always the case. And simulation, so for lack of better, you know, or oil paintings, you know, things like that. So that's the next best thing. But simulations are really informative, and usually pretty much every paper we have has a simulation component just to kind of play out what happens. So that's the best we can do.
Pancho? Oh, so following on that, is there any like open source like data sets for the MSI data, just like the ResNet, this kind of stuff? Like a really big data set for model reading?
So there's Metaspace, right? So Metaspace has quite a bit. However, we find that like very rarely it has what we need for the specific projects. Like for example, this multi-tissue, multi-replicate studies, there's very little of that, or multimodality, very little. But in terms of just data to train, I don't know. So Metaspace was designed for annotations, right? So in terms of training annotations, there will be a lot of data.
It's like you just said you are assuming the multimodality method. Multimodality, I assume people need a lot of like connecting data in order to do the click stuff.
Exactly. And so currently the way we operate really is just by collaboration, because it's very hard to have the data. So I've seen some people like use Pride, for example, massive to download, to upload mass spectrometry imaging data, but very rarely it will be the type of a design and external validation and multimodal, you know, and whatnot. Very rarely you would find this just randomly. Unfortunately, I really wish that kind of in time, there will be more of a culture of providing more data in a way that is, you know, interoperable and publicly accessible and all of that. For now, we just work one by one.
We have another question online.
Is there any evidence that some of these tissue analytes follow similar patterns of change in blood samples?
Oh,
Woohoo. I haven't done anything like that with imaging specifically, but we certainly did a lot of that in proteomics, right? And so what you can do tissue analysis to see what things are changing, and then do another kind of step to see which of those analytes actually leaking into blood or plasma. Then you can kind of try to design targeted assays to see how many of them are actually detectable and quantifiable reliably. And it kind of narrows down by a lot. And by the way, when I say we did the computational work, right? So the experimental part was obviously done by our collaborators. So this is actually quite common in proteomics. I have not seen mass spectrometry imaging experiments like that, but you know, chances are they exist. I just wasn't involved in that.
all right. So I don't see any other burning questions. If you would like to speak with the speaker or talk to the speaker, meet me after. Thank you again.
Thank you very much.